Introduction. Hello everyone, and welcome to today's lecture on the fascinating topic of fat metabolism. In this lecture, we will delve into the intricate molecular mechanisms behind lipolysis, adipogenesis, lipogenesis, beta oxidation, and various other aspects of fat metabolism. Understanding these processes is crucial for gaining insights into metabolic diseases such as obesity, diabetes, and cardiovascular disorders. So, let us begin our journey into the world of fat metabolism with the process of lipolysis. Lipolysis is the breakdown of triglycerides stored in fat cells, also known as adipocytes, into free fatty acids, FFAs, and glycerol. This process is primarily regulated by the enzyme hormone-sensitive lipase, HSL, which is activated by hormonal signals, such as catecholamines and glucagon. Upon stimulation, HSL is phosphorylated and translocates from the cytoplasm to the surface of lipid droplets within the adipocyte. Once attached to the lipid droplet, HSL hydrolyzes the triglycerides into FFAs and glycerol, which are then released into the bloodstream. Lipolysis provides a readily available energy source during fasting or periods of increased energy demand. The released FFAs are transported to various tissues, such as skeletal muscle and liver, where they can undergo further metabolism. Now that we have explored lipolysis, let us move on to the next topic of our lecture, adipogenesis. Adipogenesis is the process by which preadipocytes, undifferentiated cells present in adipose tissue, differentiate into mature adipocytes. This intricate process involves a complex network of transcription factors such as peroxisome proliferator activated receptor gamma, PreGamma, C-SOT/enhancer binding proteins, C/EBP alpha, beta, and delta, and sterol regulatory element binding protein 1C, SREB1C. During adipogenesis, various growth factors and signaling pathways induce the expression of these transcription factors. The activated PreGamma plays a crucial role in orchestrating the expression of adipocyte-specific genes involved in lipid metabolism, such as fatty acid binding protein 4, FABP4, and adiponectin. The differentiation of preadipocytes involves the sequential activation of transcription factors, cellular morphological changes, storage of lipids and lipid droplets, and regulatory changes in gene expression profiles. This comprehensive process ensures the accumulation of fully functional adipocytes capable of energy storage. Now that we have explored the fascinating process of adipogenesis, let us move on to our next topic, lipogenesis. Lipogenesis is the process of making new fatty acids from glucose or carbohydrates. It mainly occurs in the liver and adipose tissue. Several enzymes, such as acetyl-CoA carboxylase, fatty acid synthase, and stearoyl-CoA desaturase, control this process. Acetyl-CoA is used as the building block for fatty acid synthesis. Fatty acid synthase combines and reduces the molecules to create palmitic acid, and stearoyl-CoA desaturase adds double bonds to generate unsaturated fatty acids. Lipogenesis is important for membrane synthesis, energy storage, and signaling molecules. Imbalances can lead to obesity and fatty liver disease. Next, we will discuss beta oxidation. Beta oxidation is the process by which fatty acids are broken down into acetyl-CoA units for energy production. This vital process occurs in the mitochondria of various tissues, including skeletal muscle, liver, and heart. The first step of beta oxidation involves the activation of fatty acids by fatty acyl-CoA synthetase, resulting in the formation of fatty acyl-CoA molecules. These molecules then undergo a cyclical series of reactions involving oxidation, hydration, oxidation, and teolysis, leading to the production of acetyl-CoA units. The acetyl-CoA units generated through beta oxidation can directly enter the citric acid cycle, also known as the Krebs cycle, to generate ATP or be used for the synthesis of ketone bodies. Beta oxidation is a vital energy-producing pathway, particularly during times of prolonged fasting or intense exercise when glucose availability is limited. Now that we have explored beta oxidation, let us delve into the intricate relationship between insulin signaling and lipogenesis. Insulin signaling plays a crucial role in the regulation of lipogenesis, primarily in adipose tissue and liver. Insulin, a hormone secreted by the pancreas, exerts its effects by binding to its receptor on the surface of target cells. The activation of the insulin receptor triggers a signaling cascade that ultimately leads to the translocation of GLUT4 glucose transporters to the plasma membrane. This facilitates the uptake of glucose into the cell, providing the necessary substrate for lipogenesis. Insulin also stimulates the transcription of key lipogenic genes, including acetyl-CoA carboxylase, ACC, and fatty acid synthase, FAS, through the activation of transcription factors such as SREB1C and carbohydrate responsive element binding protein, CHREP. However, insulin resistance, a hallmark of type 2 diabetes, impairs the ability of insulin to effectively stimulate lipogenesis. This disruption in insulin signaling leads to dysregulated fatty acid synthesis and contributes to the development of various metabolic disorders. Now that we have explored insulin signaling and lipogenesis, let us move on to our next topic, the formation and function of lipid droplets in adipocytes. In this section, we learned about the formation and function of lipid droplets in adipocytes. These organelles are formed through the fusion of neutral lipid droplet precursors with the endoplasmic reticulum. Proteins like perilipins coat the surface of the lipid droplets and regulate their interaction with other cellular components. Once formed, lipid droplets store triacylglycerols and cholesterol esters and sequester lipotoxic species and lipolytic enzymes. Proper regulation of lipid droplet dynamics is important for maintaining energy balance and preventing metabolic disorders. Next, we will discuss the hormonal regulation of fat metabolism. Fat metabolism is tightly regulated by various hormonal signals that influence lipid mobilization, storage, and utilization. Hormones such as insulin, glucagon, catecholamines, and growth hormone play crucial roles in modulating these processes. Insulin promotes lipid synthesis and storage by activating lipogenesis and inhibiting lipolysis. Conversely, glucagon and catecholamines, in response to low glucose availability or stress, stimulate lipolysis by activating hormone-sensitive lipase, HSL. Growth hormone acts to enhance lipolysis and lipid oxidation in adipose tissue, liver, and other tissues. It also promotes the release of FFAs into the bloodstream, providing an energy source for peripheral tissues during periods of energy demand. These hormonal signals interact intricately and depend on the nutritional status and energy demands of the organism. Dysregulation in hormonal signaling can lead to metabolic disorders such as obesity, insulin resistance, and dyslipidemia. Now that we have explored the hormonal regulation of fat metabolism, let us move on to our next topic, transcription factors in fat metabolism. Transcription factors are important molecules in regulating genes involved in fat metabolism. Peroxisome proliferator activated receptor gamma, PRGamma, controls adipocyte differentiation and lipid storage. C-SOT/enhancer binding proteins, C/EBPs, promote adipogenesis and regulate lipid metabolism genes. Sterol regulatory element binding protein 1C, SREP1C, regulates lipogenesis, contributing to fatty acid synthesis. These transcription factors work together to maintain energy balance in adipocytes. In our next topic, we will discuss the role of AMPK signaling in fat metabolism. AMP-activated protein kinase, AMPK, is a critical energy sensing enzyme that plays a crucial role in regulating cellular energy homeostasis. It is activated when cellular energy levels are low, such as during calorie restriction or exercise. AMPK exerts potent effects on fat metabolism by phosphorylating and modulating the activity of various enzymes and transcription factors. It stimulates fatty acid oxidation through the activation of enzymes such as carnitine palmitoyltransferase I, CPT1, and inhibits fatty acid synthesis by inhibiting ACC and FAS. Additionally, AMPK can promote lipolysis by phosphorylating and activating hormone-sensitive lipase, HSL. It also enhances mitochondrial biogenesis and oxidative capacity to maximize energy production from fatty acid oxidation. The dysregulation of AMPK signaling has been associated with metabolic disorders such as obesity, insulin resistance, and fatty liver disease. Therefore, understanding the intricate role of AMPK in fat metabolism is crucial for the development of potential therapeutic interventions. Now that we have explored the role of AMPK signaling in fat metabolism, let us move on to our next topic, noncoding RNAs in fat metabolism. In this section, we will explore the role of noncoding RNAs, specifically microRNAs, miRNAs, and long non-coding RNAs, LNCRNAs, in regulating fat metabolism. miRNAs can bind to messenger RNA molecules and degrade them or prevent their translation, thus influencing processes like adipocyte differentiation and lipolysis. For instance, miR27A/B inhibits adipocyte differentiation by targeting PRGamma, while miR143 and miR223 promote lipolysis. LNCRNAs, on the other hand, regulate gene expression at the transcriptional and post-transcriptional levels, including adipogenesis and lipid metabolism. This emerging field offers potential therapeutic targets for metabolic disorders. Lipid rafts, specialized microdomains in cellular membranes, have important roles in regulating fat metabolism. They help organize specific signaling molecules and coordinate their functions. In fat metabolism, lipid rafts are involved in insulin signaling, lipid droplet formation, and the function of lipolytic enzymes. Insulin receptor signaling is enhanced within lipid rafts, regulating lipogenesis and glucose metabolism. Lipolytic enzymes like hormone-sensitive lipase and adipose triglyceride lipase are recruited and activated in lipid rafts during lipolysis. Finally, lipid rafts help organize and transport proteins associated with lipid droplets, contributing to their dynamics and functionality. Adipokines are signaling molecules secreted by adipose tissue that regulate energy metabolism and systemic processes. Adiponectin is an insulin-sensitizing adipokine regulated by progamma and AMPK signaling. Leptin regulates body weight and appetite through the hypothalamus. Adipose tissue also secretes TNF-alpha, IL-6, resistin, and visfatin, contributing to metabolic function and inflammation. Understanding the regulation of adipokine secretion is crucial in understanding fat metabolism. Fat metabolism is an intricately regulated process involving various steps and molecules. From breaking down triglycerides to storing and utilizing lipids, processes like beta oxidation and insulin signaling are vital for energy production. Transcription factors, non-coding RNAs, lipid rafts, and adipokines further add complexity. Understanding fat metabolism helps us comprehend metabolic disorders and develop treatments. Thank you for your attention, and I hope this lecture on fat metabolism provides a strong foundation for your future studies and practice.